So what is histopathology or histology? This is the study of the microscopic architecture of diseased tissue. We study this to find and define differences in sort of like the features of the tissue, so the integrity, cell-to-cell interactions, proteins expressions, if there's been cell invasion or damage, and the cell morphology. And we look at this by using a variety of histological techniques. So, we can have an example here. So, say you've got a patient comes in, she undergoes breast cancer surgery, so she has a small biopsy taken for pathological analysis. When the specialist reviews it, they decide they want to have DNA analysis done to look for the BRCA1 gene. So this is associated with an increased risk of breast and ovarian cancer, and then a pathohistological analysis, so the physical characteristics of the sample, so how does the tissue actually compare to a healthy sample. And how would we do this? So the first thing we have to do is obviously take the sample. The tissue's then immediately placed into transport solution, which is usually PBS or a custom medium that's sort of supplemented so that the tissue survives the transport. It's then shipped in like an express service at ambient temperatures in secure packaging to whatever lab that it's going to be tested at. When the parcel then arrives like the following day or like later on in the day at the testing lab, you need to sort of inspect the package to check that it's not been tampered with and like it's not been swapped about. And then you need to unpack the sample and then immediately input the sample ID into the database, because you can't be referring to samples obviously by patient name, and you can't have the patient name visible due to confidentiality reasons. And then in this case, because they wanted the genetic analysis done for the BRCA1 gene and they want the pathohistological analysis done, the sample needs to be cut in two. So when they're in the lab, in a sterile area, the sample sort of cut in two. It's continually submerged in a solution during this time because they don't want it to dry out. One half can be frozen for later DNA analysis and extraction. So it's frozen at about -80 degrees C. And the other half is then placed into a histocassette and fixation agent is added. This preserves the sample so that it's as it was when it was sort of alive, and this agent is usually 10% buffered formalin. The amount of fixative agent used depends on the thickness of the sample. So obviously if you have a thicker sample size, then you leave it in the fixative for longer. And at the same time, you also have to label the histocassette so you know what's sort of in it. You can only do this with pencil because the processes that it goes through later sort of washes off pen ink, so you can only use pencil. When it's been fixed, it then has to go through processing. So this used to be done by hand, but now there's like huge machines that you can that are like automated tissue processors. So it moves the cassette from the formalin solution through an ethanol series from low to high concentration to dehydrate the tissue. Once it goes through the ethanol series, it then moves into xylene, which is sort of dangerous, and then it's moved into liquid paraffin, so wax, and then it's taken out and allowed to solidify. And once it's gone through all this processing, the sample's like fully dehydrated and there's no water left in it at all. And the paraffin embedding is usually done in an embedding station, so like it has like a reservoir and paraffin. You sort of hold the sample in there, put the paraffin in it so that it's orientated the right way and let it solidify. When it's fully solidified in the mould, then you go to a microtome. So you have like, after it's been embedded in the the mould like makes it into like a square shape with the tissue sample inside. So you've got like a big chunk of wax with a tiny sample inside that you need to then get put on a slide. So you need to use a microtome to sort of slice the sections to about a 3 to 7 micrometer thickness, depending on obviously again what tissue it is and then what your sort of analysis you're doing. So you sort of use the microtome to slice it. It's a very delicate procedure. There's loads of videos on YouTube if you just sort of Google microtome. It's like a wheel crank that you have to use, and I was terrible. All my samples ripped. So yeah, and then when you have enough slices, and you can see a sample in it clearly, you transfer them into like a water bath to stretch it out, and then you can sort of scoop it up with a slide. You then label the slides in pencil, and voila, you have a biopsy sample on a slide for you to look down a microscope at. It's also very important to remember that this is human tissue, so the Human Tissue Act stands for its use. But diagnostic and forensic procedures tend to have special exceptions from the HTA rules. When you and then when you have the slides, you have to do, obviously it's sort of like a see-through slide, so you have to do something called staining. It tends to be H&E, so I can never say them, hematoxylin and eosin. So the hematoxylin, I think it is, and sort of stains the acidic structures blue, so the nuclei and sort of things like that, and the eosin stains more basic structures to pink. There's loads of images that you can find online and stuff.